There is a secret written inside the DNA of Black Americans, a secret so medically consequential that without it, modern medicine as we know it would not exist. In 2024, scientists made a discovery that should have been front-page news everywhere. The National Institutes of Health sequenced the genomes of nearly 250,000 Americans through a program called All of Us. And inside the DNA of Black participants, they found 275 million genetic variants that had never been documented in medical science, ever. And that was just the beginning. What researchers have been piecing together for decades is far bigger than one study. Black Americans carry ancient genetic adaptations, forged over 300,000 years of African evolution, that became the direct biological foundation for some of the most important medical breakthroughs in history. The first CRISPR cure ever approved, the drug that blocks HIV from entering human cells, treatments for kidney disease affecting millions. All of it traces back to DNA that has been sitting inside Black Americans this entire time. Before we dive in, comment where you are watching from and click subscribe. Let's start with a number that stops most people cold: 90%. For decades, right up until the early 2020s, over 90% of participants in large-scale genomic studies were of European descent. 9 out of every 10 people whose DNA was used to build the foundational databases of modern medicine were white. Think about what that means. When scientists were figuring out which genes cause which diseases, which variants indicate drug risk, which markers predict outcomes, they were working from a picture of human biology that was almost entirely European. They were reading one chapter of a 50-chapter book and calling it the complete story. Dr. Josh Denny, Chief Executive Officer of the All of Us research program, put it plainly: the lack of diversity in genomic research directly hindered scientific discovery, not slowed it, hindered it. Answers were always there, written in DNA, that medicine simply couldn't see because it wasn't looking at the right people. That absence had real consequences: drugs that didn't work correctly, doses calibrated wrong, diseases misdiagnosed because the reference data didn't apply to Black bodies. For generations, Black Americans were effectively invisible in the databases doctors used to make decisions. But what that absence also means, and this is what science is only now grasping, is that within the DNA of Black Americans sits an enormous, largely unexplored vault of genetic knowledge, a vault locked for decades. Scientists are finally cracking it open. To understand what's inside that vault, you have to understand something about Africa that most people never learned in school. Africa is not just the birthplace of humanity. It is the world's greatest reservoir of human genetic diversity. Modern humans have existed in Africa for approximately 300,000 years. Populations lived, diversified, and migrated across the continent, and they adapted to radically different environments: the Sahara, the Congo rainforest, the East African savanna, and the coastal West African tropics. Each environment applied different evolutionary pressures, different parasites, different diseases. And over hundreds of thousands of years, African genomes responded by evolving solutions. Then roughly 60,000 years ago, a small group of humans migrated out of Africa and eventually populated the rest of the planet. Every non-African person alive today descends from that group. Every population outside Africa, Europeans, Asians, Indigenous Americans, represents a genetic subset of African diversity, a fraction of the variation that exists in Africa. Scientists call this the founder effect. Imagine Africa is a library with 10,000 books. The group that left took a backpack. Everyone outside Africa descends from the books in that backpack. Africa still has the entire library. This is why when geneticists study populations for diversity, Africa wins by an enormous margin, and Black Americans, descending primarily from West and Central African populations, carry a significant portion of that library within them. Let's talk about one of the most dramatic examples in the history of genetic medicine. It starts with a disease that has killed more human beings than any other in recorded history: malaria. The Plasmodium parasite has been killing humans for at least 100,000 years. In Africa, where it has been endemic longer than anywhere else, it was the single most powerful evolutionary force shaping the African genome. And the African genome fought back. Over tens of thousands of years, natural selection produced multiple genetic adaptations specifically designed to resist Plasmodium. These are among the most medically significant genetic variants ever discovered. The most well-known is the sickle cell variant, a single mutation in the hemoglobin gene known as HBB. In its homozygous form, it causes sickle cell disease. But in its heterozygous form, with one copy, it provides a tenfold reduction in the risk of severe malaria. 1 in 12 Black Americans carries the sickle cell trait. For centuries, it was only seen as a disease, a curse written in DNA. But medicine has slowly come to understand that this mutation is an ancient weapon, so effective against one of humanity's deadliest killers, that it persisted in the population despite its costs. Studying that mutation changed the world. The sickle cell mutation was the first human disease ever traced to a specific molecular defect. Everything modern medicine knows about hemoglobin genetics, every drug, every diagnostic tool, every treatment protocol, traces back to work done on a variant that exists almost exclusively in people of African ancestry. The second major malaria adaptation is less well known, but arguably more extraordinary. It involves a protein called the Duffy antigen, also known as DART. The Duffy antigen is a receptor that sits on the surface of red blood cells. It plays a role in regulating inflammation. For the Plasmodium vivax parasite, it is the doorway. Vivax malaria requires the Duffy antigen to enter where red blood cells. Without it, the parasite cannot infect. Approximately 42,000 years ago, in West African populations, a mutation occurred in the promoter region of the Duffy gene, a tiny molecular switch that silences the gene on red blood cells. People with this mutation, called Duffy negative, simply do not display the Duffy antigen on their red blood cells. The parasite tries to enter, but there is no door. This mutation spread rapidly through West African populations because it conferred a massive survival advantage. Today, approximately 68% of African Americans carry the Duffy negative phenotype. In certain West African populations, it approaches 100%. But here is what stunned medical scientists. The Duffy antigen does not just serve as a malarial entry point. It is also a receptor for a class of immune signaling molecules called chemokines, molecules that regulate inflammation, recruit immune cells, and play central roles in how the body responds to infection, injury, and disease. Understanding how this African genetic adaptation modulates the immune system has opened entirely new frontiers in immunology, cancer biology, organ transplantation research, and inflammatory disease. The molecular architecture of a 42,000-year-old African survival mechanism is now one of the most intensely studied systems in modern biomedical research. If this is the kind of deep science and hidden history that keeps you up at night, subscribe to this channel right now. We go deep on the stories that mainstream science journalism never tells completely. Hit subscribe and turn on notifications so you do not miss what is coming next. Now let's talk about a gene that is, in 2025, at the center of one of the most urgent areas in all of medicine, the APOL1 gene. In West and Central African populations, variants of the APOL1 gene, specifically called G1 and G2, evolved as a direct response to a parasite called Trypanosoma brucei, the organism responsible for African sleeping sickness. Sleeping sickness is a devastating, frequently fatal neurological disease transmitted by the tsetse fly. The G1 and G2 variants encode a modified protein that can kill Trypanosoma brucei. People who carry these variants are significantly protected against sleeping sickness, a highly specific genetic weapon forged against a highly specific pathogen. But here is the complication. The same variants that protect against sleeping sickness increase the risk of kidney disease, specifically APOL1-mediated nephropathy, which can progress to end-stage renal failure. In the United States, Black Americans carry a dramatically elevated risk of kidney failure compared to white Americans. For years, medicine explained this as a racial disparity without understanding the mechanism. A POL1 research revealed the truth. The biological mechanism is not race. It is an ancient adaptive variant selected for survival in an environment where sleeping sickness was a leading cause of death. Right now, in 2025, pharmaceutical companies are running clinical trials for drugs specifically targeting APOL1-mediated kidney disease. Drugs that could not exist without the foundational understanding of what this gene does and why. That understanding came directly from studying a variant found almost exclusively in people of West and Central African ancestry, the kidney disease killing Black Americans in disproportionate numbers. The medicine being developed to stop it. Both trace back to the same ancient African genetic adaptation. The same DNA that served as a survival weapon for millennia is now the key to a cure. There is another chapter, and it involves a disease the entire world is still fighting: HIV. In the 1990s, scientists discovered that certain people appeared essentially immune to HIV-1, not resistant, immune, exposed repeatedly and still uninfected. The explanation was a genetic variant called CCR5 delta 32, a deletion of 32 base pairs in the CCR5 gene. HIV-1 needs 2 entry points to infect a cell, the CD4 receptor and CCR5. People with 2 copies of CCR5 delta 32 produce a non-functional CCR5 protein. HIV literally cannot find a door. This gave scientists a precise molecular target, exactly how HIV enters. So that target powered an entire class of antiretroviral drugs. The FDA-approved drug Malaria works by blocking CCR5, mimicking this natural resistance. The CCR5 delta 32 variant is found almost exclusively in people of European descent. It is virtually absent in African populations. But African populations carry their own distinct CCR5 variants that affect HIV progression in ways medicine is still working to understand. The critical insight came from comparing these populations. By studying the differences between African and non-African CCR5 genetics, scientists gained a complete understanding of how this receptor works and how it could be therapeutically targeted. The contrast itself was the discovery. In 2009, a man known as the Berlin patient received a bone marrow transplant from a donor with CCR5 delta 32 and became the first person in history functionally cured of HIV. Scientists are now working to replicate this with gene editing. The entire field is building toward a genetic cure, and the conceptual origin lies in studying resistance variants across human populations. Both African and non-African genetic diversity were essential to that roadmap. And now we arrive at the single most consequential moment yet in the story of African DNA and modern medicine. December 2023, the FDA approved 2 gene therapies for sickle cell disease. The first, Casgevy, was not just a treatment. It was the first therapy approved by the FDA in human history to use CRISPR-Cas9 gene editing. It was the first time this revolutionary tool was authorized inside a living human being to correct a genetic disease. Think about that. The first disease ever treated with CRISPR was sickle cell disease. It is a disease that exists almost exclusively in people of African and African American ancestry. The mutation Black Americans have carried for tens of thousands of years. First studied, first molecularly characterized, first understood as a genetic disease, became the proving ground for the most powerful genetic medicine tool ever created. The first person in the United States to undergo CRISPR gene editing for sickle cell was a Black American woman named Victoria Gray. In 2019, she received the experimental treatment. After a lifetime of debilitating pain, she became pain-free. When Casgevy was approved in 2023, she described it as a blessing. The FDA also approved a second therapy called Lyfgenia. In December 2024, a patient who received it was described by doctors as cured, not functionally managed. cured. For most of the past century, the only potential cure for sickle cell disease was a bone marrow transplant requiring a perfectly matched donor, something most patients could never find. The average life expectancy was approximately 52 years old. There are credible voices who argue that the disease's disproportionate impact on Black Americans contributed to decades of underfunding. They say a disease affecting primarily white patients would have received gene therapy a generation earlier. But here is what cannot be argued. The deep scientific understanding of the sickle cell mutation, forged in African populations against malaria, studied for over a century in Black American patients. The first disease ever linked to a specific molecular defect made CRISPR's first clinical application possible. The genetic heritage of Black Americans didn't just provide a target, it provided the proof of concept that made CRISPR medicine real. In 2024, the All of Us research program confirmed what geneticists had long suspected. The team sequenced whole genomes of 245,388 Americans, 46% from underrepresented minorities. Among more than 1 billion genetic variants identified, 275 million had never been documented in scientific literature. For every disease studied, all 117 conditions, from cardiovascular disease to diabetes to cancer, there are now genetic associations in people of African ancestry that were invisible before. Not because they didn't exist, because no one had looked. Researchers also found something immediately clinically relevant. A genetic variant had been causing thousands of Black patients to undergo unnecessary bone marrow biopsies because their white blood cell counts were lower than the normal range calibrated to European populations. It turned out to be a benign, common variant in people of African ancestry, not disease. Normal Black DNA had been misread as pathology for decades. This happened because the reference data was built almost entirely on European genomes. Real patients underwent painful procedures because medicine didn't have the right data for their DNA. That is changing now. There is a broader truth buried in all of this that deserves to be said directly. Modern medicine built its understanding of human biology on a narrow slice of genetics, and that slice was called universal. It was not universal, it was European. For generations, the adaptations forged in African populations were either ignored, pathologized, or studied only in the context of the suffering they caused. Sickle cell was treated as a curse before anyone appreciated its survival logic. APOL1 variants were linked to kidney failure long before they were recognized as ancient antiparasitic weapons. The Duffy antigen was categorized as an absence before medicine understood the profound biological mechanism that absence represented. The framing was always deficit. What these populations were missing, what they were more susceptible to. The emerging science tells a different story. What African populations and the Black Americans who descend from them are carrying is the deepest, most biologically sophisticated archive of human adaptive evolution on the planet, and it has been there all along. Here's where science stands in 2025. Drugs targeting APOL1 are in active clinical trials because African DNA revealed how the kidney and immune system interact in ways no other population could have shown. CRISPR medicine is expanding rapidly with sickle cell as its first approved application and dozens of other diseases in the pipeline. Every advance builds on the molecular foundation first laid by studying hemoglobin genetics, a field born from a variant found almost exclusively in African ancestry. The Duffy antigen is now being studied in cancer biology, where evidence suggests it plays a role in tumor progression. What began as a blood group curiosity is becoming a therapeutic target. All of Us now has more than 860,000 participants enrolled, generating new variants and medically actionable findings every month. Findings that will, for the first time, genuinely apply to Black Americans as patients. The genetic vault ignored for generations is now the most valuable resource in all of medicine. Victoria Gray, a Black American woman from Mississippi, spent her adult life managing pain most people cannot imagine. Bone crises, exhaustion, hospitalizations, watching her own body fail her. In 2019, she volunteered for a clinical trial. Scientists edited the relevant gene using CRISPR and returned the modified cells to her body. Her hemoglobin improved. Her pain crises stopped. When Casgevy was approved in December 2023, the first CRISPR therapy in history to receive FDA approval, Victoria Gray was there. She did not just benefit from the medicine. She is part of why the medicine exists. Without the sickle cell mutation, without the ancient African adaptation that caused her decades of suffering, the molecular target for CRISPR's first human therapy would not have been found. Without the research conducted on Black patients that mapped the hemoglobin gene and built the entire field of molecular hematology, the tool to edit that gene would not exist. Her DNA and the DNA of every Black American who carries the sickle cell trait is woven into one of medicine's greatest achievements. That story should have been told long before she spent decades in pain, but it is being told now. The story of Black Americans and DNA is not a story of vulnerability. It is a story of survival, of an immune system forged over 300,000 years against the most dangerous pathogens on Earth, of genetic adaptation serving in the 21st century as the molecular templates for the most advanced medicines in human history. The next time you hear about a CRISPR cure, remember where the science started. The next time you hear about precision medicine, remember whose DNA was missing. The code saving lives right now was written in African bodies long before medicine knew it was looking. The vault is open, and what's inside is going to change everything. If this video changed how you see genetics and history, share it with someone who needs to hear it. Subscribe if you haven't already.